careful with the bandwidth. If you can change your Zoom name, if you have a name like, I don't know, iPad 101 or something, it's better, please, if you can change your Zoom name so that you have your name on it so we can see who you are. If you have some problems, technical problems, it's always best to close and restart the Zoom and then to start again. Our colleagues from IITA are running the Zoom part of it, and if you have any problems, just post something in the chat and they will come back to help you. We will be using the chat a lot. I'm assuming that almost everybody here will be a Zoom expert nowadays. We're all Zooming around the world. So you maybe know, but if you're not familiar with the chat, there is the chat, and you should be able, we'll be using the chat quite a lot. We'll be posting announcements, we'll be posting process updates, and we'll be asking you to post your questions and answers as well in the chat. So that's going to be important for you. Where are we on time? I think let's already get started. What I'd like you to do is to just so we warm ourselves up and we know roughly who's here and roughly why you're here. Perhaps you can post in the chat, please. I'm going to post my message here in the chat. We're looking for, I know that not everybody is here yet, but let's already start by posting a chat message. I'm trying to post the chat message myself, but for some reason it's not coming properly. 1 second. So I'm trying to post the chat. Yeah, thank you very much, Lima. So what I'd like you to do is post something post a quick message. Tell us who you are. I don't seem able to post in the chat for some reason. Oh, okay, so post in the chat, give us your name, give us your affiliation, and maybe you can tell us why is plant germplasm health important to you? What is it about plant germplasm health that's important? So I've just posted my question in the chat. Oops. Yeah, why is plant germplasm? Oops, that's the wrong chat message here. Good, yeah. So why is plant germplasm health important to you? Good, tell us who you are. Post quickly so we can see where people are coming from. We're seeing Thailand, Pakistan, Lima, Germany, Chiang Mai, Indonesia. Okay, a very broad crowd from all over the place. CAB International, Philippines, excellent. So if you can just post a very brief, just post something before we really get going. I see we're up to 173 people. I'm looking to the organizers. Are we ready? Are we ready to move on? I think we just go with who's here. And as people come and join, they can come and join us. Yes. So Abu Hai from the Philippines, great. India, Malaysia, Mexico, super. I know there's different time zones. So we're happy to see you. Macedonia, Uganda, super. A good, broad distribution of people. So we're from all over. So let me just go on. I'm going to get into the welcome part here. So just to remind you, in case you're not aware, this is the second in a series of 4 webinars. The first one happened in January about climate change and plant health. Today, we're really focusing on germplasm health and particularly how we can avoid the spread of transboundary diseases. What we're trying to do today is to really inform you. So we have a great bunch of speakers. They're going to be informing you on some of the big challenges, solutions, opportunities. They'll be illustrating. We hope they will inspire you. And we're going to be hopefully facilitating some interaction among you. And we will be primarily using the chat for that interaction. So I will explain that as we go through. There is a social media campaign behind all of this. You can see the hashtags on my screen. And if you go to Twitter, you'll see a lot of reporting going on, and there'll be some live reporting and sharing of information from the meeting. So let me go on without further ado. What should you expect? We will have a keynote, which will set out the big issues. As people speak, as Safa presents her keynote, we want you really to be active in your listening and active in your interaction. Please use the chat, post comments, post queries, post reactions, share your experiences. You know, let's have a conversation around the chat as we hear the speakers as we move along. We'll have a roundtable panel. We've got 5 people from different organizations sharing their perspectives on different challenges in this area. We'll be doing some interaction and reflecting. And just want to have a special welcome to our YouTube audience. This is also being live streamed, and we will be, hopefully you may also be able to post your comments and queries. Everything is recorded. Everything will be available afterwards. Don't worry about getting copies of slides. Okay, in the chat, just to remind you, we're going to, you're welcome to use the chat. We really appreciate your active contributions. In the first webinar, some people were demanding that they have an attendance certificate. For this webinar, there will be no attendance certificates in advance. We don't really think it's an appropriate place to apply for jobs in the chat. So please be respectful, interact, engage, respect the topic, respect the speakers, and let's use the opportunity to learn and engage. So just as a piece of continuity, the first webinar was, I think, 2 or 3 weeks ago. We were looking at climate change and plant health. And our moderator then, Sonja Vermeulen from the CGIAR System Management Organization, she pulled together 5 key takeaways. One, that with climate change coming, this is really, really important and urgent issues, challenges. We really have to be ambitious to tackle them. She emphasized, these were messages that she drew out of the conversations and interactions. The need for cooperation and coordination, resource sharing, scientists, researchers, we need to be co-defining the questions and developing solutions with the users, not just keeping it as a research, talking to research conversation. We're going to look for opportunities to engage with the private sector, civil society, and really get empowered. She really talked about, so those are the 5 points that came out of the first one. I'm going to very briefly just run through who the webinar organizers are. This is an effort cutting across different CGIAR organizations, but also partners beyond the CGIAR, just to draw your attention to these 4 people who made everything happen. And you will be hearing from 3 of the 4 as we move through today. Vivian Atakos is keeping herself in the background, but she has been setting up all the communication efforts behind this. So that's who the organizers are. What's our agenda? We're now I'm just about to hand over to Jan Kreuzer, who's going to give us a welcome. We're going to share a video with you. We have a great keynote, then that interactive roundtable. You're invited to contribute. We'll have a kind of a second part of the roundtable, and then we'll wrap up everything. We should finish by 13:30 London time, or Scotland time is where I am. So without further ado, I'm going to hand over to Jan. I'm going to stop sharing my screen. And Jan is currently, I'm just going to stop sharing here. Jan is working at the International Potato Center, where he's the head of the Crop and System Sciences Division. He also heads ZIP's Germplasm Health and Quarantine Unit. He's a plant breeder and a virologist. He's been working at ZIP for quite a few years, involved in biotechnology, plant pathology, and diagnostics research. So I'm going to stop sharing. I'm going to hand over to Jan, who will welcome you all and tell you why we're here. And then we will show a video. Jan.
Thank you. You can hear me?
Perfect.
Great. Okay, so good morning, good afternoon, or good evening, everyone, depending on which part of the world you're connecting from. As you can see, it's morning here and the sun is just about coming up. So welcome to the second session of the CGIAR International Year of Plant Health Seminar Series, which is dedicated specifically to germplasm health. This might beg the question as to what is germplasm and how is it different from any other plant health. Put in a simple way, germplasm refers to source plants, either source of a specific cultivar with specific combinations of traits from which plants will be further multiplied for cultivation, or as sources of unique genes and traits that can be used by breeders to create new and improved cultivars. And because these are source plants, they are often sent around internationally, they trade internationally, they go in small quantities and are subsequently multiplied and used in the importing countries. And that explains, of course, the importance of these source plants, because if the source plant is not healthy, then whatever you multiply or use it for is probably also going to have health problems. And germplasm with unique traits often also come from the region of origin of a species, which is likely also the region of origin of its pests and diseases, which provides an additional risk of introducing these into imported countries if you don't take care to make sure that your germplasm is really completely healthy. Because the CGIAR gene banks contribute to over 80% of recorded germplasm transfers globally, they have a specific responsibility in this aspect to ensure that transfer occurs safely. And this is regulated by the CGIAR germplasm health units, which will be presented in the keynote lecture, together with the challenges faced and opportunities, and after which we will have discussions about germplasm health. But before I introduce our keynote speaker, we will start out with a short video introducing the importance of germplasm health and for sustainable global future in agriculture. So if I can ask Tunde to start out the video.
CGIAR germplasm health units in the prevention of transboundary pests. Plant pests and diseases threaten our global food security, resulting in up to 40% food crop losses annually, $220 billion in agricultural trade losses, decimated livelihoods, and millions left in hunger. Climate change further threatens to reduce crop yields. Rising temperatures are increasing the incidence of pests and diseases. Global food security is under threat at a time when the population is expected to reach 10 billion by 2050. Now more than ever, conservation and access to crop agrobiodiversity are essential in addressing agricultural pressures in a climate-changing world. But transporting germplasm requires careful handling to mitigate the spread of pests from one location to another. To address the hazards pests pose to our food systems and agrobiodiversity, CGIAR centers establish germplasm health units. GHUs have 3 objectives: avert the spread of quarantine pests with CGIAR germplasm transfers, prevent pest outbreaks, and safeguard biodiversity. GHUs are the gateways for safe and efficient germplasm exchange. They ensure compliance with the FAO International Plant Protection Convention and national phytosanitary regulations. More than 100 countries reached in 2018 and 2019 with germplasm transfer from gene banks and breeding programs. More than 453,000 samples tested. 6% were found to be pest affected and eliminated. To control the transboundary spread of pests, $12 million is spent annually on the generation of clean germplasm and preventative diagnostic testing. These efforts allow for germplasm transfers that would be impossible without proper testing. Access to germplasm helps fuel breeding programs, generates income all along the value chain, grows farmer resilience to climate change, and helps feed a hungry world.
Okay, thank you. Can I have the next slide, Peter? Okay, great. So now it is my pleasure to introduce our keynote speaker, Safa Kumari, who will make a presentation on behalf of the GHUs of the 11 CGIAR centers which hold plant germplasm. Safa is an accomplished scientist and, like me, a plant virologist by training, and she heads the Seed Health Lab at ICARDA. Currently, she's based at Terbol station in Zaleh, Lebanon, after risking her life to save ICARDA germplasm from her home country, Syria, when the conflict erupted. She was named one of BBC's 100 Women of the Year in 2020, and she will be presenting now on the germplasm health unit's work in the CGIAR centers. So please, Safa, the floor is yours.
Can you see my presentation?
Yes, we can. Yeah.
Okay. Thanks, Mr. Chairman. Ladies and gentlemen, good morning, good afternoon, and good evening, wherever you are. My name is Safa Kumari. I'm originally from Aleppo, Syria. As Jan mentioned, I'm working for ICARDA, and I'm handling the responsibilities of both seed health testing and virology research. I would like to thank the organizer for giving me the opportunity to join this important session, and I'm glad to be here with you today. Now, let's start it from COVID-19 pandemic that made viruses famous worldwide and turns billions of people's lives upside down and changed social interaction and how we work, travel, and communication. Many terms become now commonly used word by the public, like virus and strain, isolation, treatment, immunity, PCR test, diagnostic, vaccine, and recently mRNA, and quarantine. Unfortunately, less attention is given to plant viruses because they don't affect humans directly, even though plant viruses and other plant pests cause high impact on global food security. More than about 1,300 known pests and pathogens infect agricultural crops globally, with an estimated economic impact of around 540 billion US$ annually. Most severe damage inflicted by introduction pests and pathogens, which are serious threat to food system and biodiversity. This is the CGIAR tweet from the first series of CGIAR webinar of February 12th. The tweet say, because of disease and pest, we lose about 20 to 40% of our food crop. How many people we could feed with that lost food? So, what we can do as plant health specialists to preserve the global food system? The answer is very simple, but to reach the goal is not easy. The most important step is safe genetic resources exchange to prevent international spread of pests and pathogens. In the coming few minutes, I would like to talk on germplasm health in preventing transboundary spread of pests and pathogen. This presentation on behalf of my Germplasm Health Unit colleagues who represented 11 CGIAR centers. You will see here the name is written in different color. The names that written in red color are for ladies, and the name in blue are for gentlemen. From out of 11 GHUs are led by women. I can say we are lucky for that. And 40% of CGIAR GHUs staff, international and national, are women. Recently, GHU's group published a review paper entitled Phytosanitary Intervention for Safe Global Germplasm Exchange and the Prevention of Transboundary Pest Spread, the role of CGIAR Germplasm Health Unit. I strongly recommended to read this review paper if you are interested with this topic. CGIAR is a global partnership that united international organizations engaged in research about food security. It's carried out at 15 international centers operating in around 100 countries through a combined team of around 8,000 scientific staff. 11 CGIAR centers out of 15 have gene bank, which conserve around 770,000 accessions of cereal, grain, legume, forage, tree species, root and tuber crop, and banana. Around 85% of the international germplasm distribution from CGIAR program, and you can see from this figure, during 2008 to 2010, the CGIAR Center have distributed around 500,000 germplasm, about 23% from gene bank and 61% from other or from crop breeding program to around 160 countries. International germplasm transfer have been recognized as an important pathway for spread of pests and pathogen through human activity of collection and distribution, such as moving pests between geographic and international into new regions where they are not known to exist. The spread of pests has increased dramatically in recent years through international trade, like infected seed, tissue culture material, and others, climate forces, wind and rainfall, and insect or other vector and seed and agriculture trade. Here are some recent examples of outbreak caused by transboundary pest introduction in the region of CGIAR operation. I will not go all of them. I will just give you an example. The banana wilt caused by Fusarium oxosporium, tropical race 4, introduced from East Africa to Southern Africa and Latin America. Fall army worm was introduced to Africa and Asia from America. Potato cyst nematode outbreak in Kenya and Uganda was introduced from Europe. Germplasm Healthy Unit serve as the CGIAR centers gateway for germplasm exchange by ensuring compliance with the FAO International Plant Protection Convention procedures and the international standard for phytosanitary measures used by National Plant Protection Organization to prevent the introduction and control the spread of pests along the plant or plant product. GHU have three main objectives: avert the spread of quarantine pests in all CGIAR germplasm transfer, prevent pest outbreak, safeguard biodiversity. The role of CGIAR germplasm health unit is concentrated on develop diagnostic tool and procedure for germplasm healthy testing, develop and adopt phytosanitary procedure to generate pest-free germplasm, ensure that all movement of germplasm comply with the guideline of importing and exporting country and that material are free of quarantine pests, conducted pest risk assessment of germplasm activity, including conservation, seed increase, and transfer, organize GHU community of practice to form a network of center for transboundary pest prevention, and last activity is contribute to the phytosanitary capacity development. GHU engage with the National Plant Protection Organization of the host and recipient country to organize import permit and phytosanitary certificate, conduct inspection of regeneration field, and prepare germplasm for exportation or importation in accordance with the international standards for phytosanitary measures and other recommended action. In this slide, you can see the summary of the germplasm import protocol for CGIAR in collaboration with the MBBO. First, we started import permit request to MBBO, process germplasm as per MBBO approval procedures, laboratory healthy testing, fumigation, seed treatment, and finally growing only the acceptable material at post-entry quarantine field, followed by field inspection and other type of healthy testing. This slide summarizes the germplasm export protocol also for the CGIAR in collaboration with the MBBO against the quarantine regulation of the recipient country through field inspection, laboratory testing, fumigation, seed treatment, and all the shipment dispatched along with the required quarantine document, phytosanitary certificate issued by MBBO, and other document fulfilling the international phytosanitary regulation. The GHU routinely check for about 320 pests and pathogens that are endemic in the germplasm production site. In addition, the testing cover other pests are as listed in the import permit of the country that received germplasm. During 2018 to 2019, around 450,000 samples tested by GHUs, and 6 of them were pest affected and eliminated. More than 65,000 samples distributed to 100 countries per year. 10 to 20 million US dollar is spent annually on the generation of clean germplasm to control the transboundary spread of pests and pathogens. The key challenge of germplasm exchange are emerging new diseases and insect pests. Minor pests and diseases are becoming important due to climate change, variable standard and different phytosanitary demand, and limited funding for preventive measures. And let me conclude, GHU effort averted the inverted spread of quarantine, pest, and pathogen. GHUs enable germplasm access to global crop productivity improvement. A global distribution of GHUs in the development world enable them to play an important role as center of excellence in support of national or regional pest and disease surveillance and rapid response to limit their spread. To avoid the scenario of COVID-19 pandemic in agricultural crop, we should pay attention on the safe and efficient transfer of germplasm. Sure, this required enhanced collaboration with phytosanitary organization and research institution to obtain the most advanced information on pest detection and epidemiology. A strong case exists for positioning GHUs as part of a global network of phytosanitary hubs for research, diagnosis, and control of established and emerging pests as part of the One CGIAR program. Finally, let us take coronavirus as a good lesson and a chance to improve our facilities and infrastructures, and above all, to learn how to become more ready for the next emergency crisis. And also, we need to ask ourselves two questions. What if COVID-19 would have been contained before its global spread? And are we ready for new emerging pests and pathogen? I can say prevention is better than cure. And many thanks for your attention.
Thank you very much, Safa, for that wonderful presentation, and you're in a perfect timing as well. So let me then move ourselves on. You've been seeing my colleagues, we were asking you to post any comments and questions in the chat. I saw one question coming in. So Safa, if you have some answers, or other people, please share them in the chat. We don't really have opportunity to do a kind of a plenary conversation on with voices. Just use the chat if you can. Now, what I want you to do now is to think a little bit about what you've heard. We need to be ready, some of the big challenges that are arising, and I'm going to do a chat exercise. So what we will do here, I'm going to post in a minute a question in the chat. And as I post that question, I want you to take no more than half a minute to think about your answer to the chat, yeah? And I want you to type the answer in, but do not post it until I say go. So you have to wait for a moment. We call this a chat wave. So basically we all wait, and then we have hopefully 280 comments on the chat. So here is the question. Whoops, gone too quickly. What do you see as the most significant challenge to achieve plant germplasm health? So I would like you please to post. Think about that, take 20 seconds. And when I give you the instruction, please post your answer in the chat. And I'm looking for 280 chat comments. And Jan, I'm going to ask you to help me as we look at all these comments, just to make sense of them. So I'm going to give you a countdown from 5 to 0, and then you can post. So 5, 4, 3, 2, 1, go. Please post your answers. Good. So Jan, what are we seeing? It's a big challenge to keep up with all these ideas, huh? What are we seeing? Jan, I need help. We're seeing proper confinement, capacity building, quarantine. What are the big challenges?
Yes, there's a lot of challenges there coming up. I see politics there, diagnostics, cleanup, the big movement of material between many different regions. I guess that's the globalization, climate change, Diagnostic comes up quite a bit. Funds,
Funding.
of course, funding, always there.
Someone said they're so small you can hardly see them. Is that a big challenge, or do we already have the answers for very small things? Pathogens.
Difficult.
Difficult.
Quarantine.
Safa, this is following your Safa. Do you see anything here that is a challenge that you haven't seen before, or something that really resonates with you?
I think one of them, they mentioned phytosanitary certificate. That means the collaboration between the national program and the research institution. There is something there. It's very interesting. What he means that the problem with the phytosanitary certificate.
Okay, good.
Phytosanitary certificate is the activity or the responsibility of the National Plant Protection Organization. And also some of them, he mentioned breeders. That mean there is related to the interaction or collaboration between the plant health scientists and the breeders.
Good. Okay. So that
I
gives us
see
a
an
flavor.
interesting one here too.
Yep. Yeah. And anything else you're seeing that jumps out on you from the list?
Yes, so yeah, the lack of protocols for orphan crops when you're moving them around, I think that's an interesting point as well. We work with a lot of small crops, and other people do as well, and in many cases almost nothing is known about them.
Nothing is known about the crops, or nothing is known
about the diseases and the protocols to detect the pathogens.
Are there like orphan pathogens as well? Okay.
Exactly, orphan crops.
Okay, everybody, so you have a last chance to send something, and then we're going to move on. And yeah, so go ahead, sorry.
Oh, there seems to be a lag.
Anything else, Jan, from your side? Yeah. Good, okay, we're perfectly in the time. So what I'd like to do now is we're going to move on to the next slot. So thank you very much. We appreciate your contribution to the chat. We will do a couple more of those. We will also do a couple of polls. So if you have comments, queries, suggestions, please post them in the chat, and then the organizers and the speakers will be looking at them and responding to them. So I think, Safa, there was a specific question for you way up in the chat. You might have to dig it out if you could have a look at that one. So let me go on. So we're going to now change the focus. We've had a very nice scene setting presentation from Safa. We're going to hear some different perspectives. And we got together a group of eminent scientists to share some different perspectives around germplasm health. So we have a kind of a panel for you, a roundtable panel. This is going to be run in kind of two phases. We're going to have 5 panelists. They're going to be in conversation with Lava. And Lava is Dr. Lava Kumar, who works at IITA. He is the head of germplasm health, virology and diagnostics, and he coordinates the germplasm health component of the CGIAR gene bank platform. He's been working at IITA, the International Institute for Tropical Agriculture, for 25 years, so he knows what he's doing. So Lava will be he will be the interviewer, he will be the host of the roundtable. So we've asked each panelist to start off with a very short 3-minute pitch. Tell us about the challenge, tell us what the issue is, tell us why it's important. As we move through, we invite you to chat. We will organize a couple of polls. So we want your comments and questions and reactions, because in the second round, after we've had the first round, Lava will bring everybody back and he will moderate a Q&A conversation. So Lava, over to you. I'm going to show the list of panelists on the next slide, and I think you're going to introduce the different panelists. Lava, yes?
Yes, Peter, thanks a lot.
I'll mute myself now.
Thank you very much. So once again, warm welcome to all the participants. Good morning, good afternoon, good evening, wherever you are. We had very exciting beginning, and Safa has given a very good overview of the germplasm health work, and she also emphasized the relevance of this work in the context of all the pest and disease outbreaks that is happening around the world, and the navigating germplasm, which is a very important activity of CGIAR, and the good or the pertinent work that GHUs are doing together with the National Plant Protection Organizations. Now we will probe some of the key issues that are centered around the germplasm health policy and regulations. So we have an excellent panel who are plenty of experience, lots of experience on the issues of germplasm and the phytosanitary issues. I would invite to start the discussion. I would first start with Mr. Mirko Montuare from FAO IPPC. He is the project officer for International Year of Plant Health, and he has very rich experience in governance and institutional development, also on pest and diseases, food safety issues, network development management, and most importantly, information and knowledge management and communications. So I would ask Mirko to give his opening remarks. You have 3 minutes, Mirko.
Thanks very much, Lava. And first of all, let me start by thanking CGIAR and particularly SIP and AITA for convening this important webinar. I'm very grateful to be part of such a distinguished panel of speakers representing the Secretariat of the International Plant Protection Convention at the occasion of the International Year of Plant Health. So first of all, germplasm is essential for biodiversity conservation and research. Yet, like other plants and plant products, and as it was wisely explained by the previous speaker, Dr. Kumari, like other plants and plant products, germplasm may carry pests and diseases or pathogens. So the work of the CGIAR Germplasm Health Unit is actually key to sustain plant health, and the work of the International Plant Protection Convention and also national plant protection organizations, especially developing countries and those hosting biodiversity hotspots. So I made here some examples, like gene bank standards for plant genetic resources for food and agriculture were developed by the FAO's International Treaty on plant genetic resources for food and agriculture in 2013. And they basically set the benchmark and support the key international policy instruments for the conservation and use of plant genetic resources. These standards actually describe management practices for ex situ conservation in seed banks, for field gene banks, and also for in vitro and cryopreservation. When it comes to the international transfer of germplasm, so at this point, international and national phytosanitary regulations come into play. And I have to say that CGIAR and other research organizations appear to be making, as we see, the best use of available methods for international transfer. I reported here some examples, like ISPN 36 on integrated measures for plants for planting, as well as ISPN 38 on international movement of seeds, which address some of the challenges we have with the international movement of germplasm. But of course, we can expect more on this with the work that is being done on commodity standards, for example, which is highlighted in the IPPC Strategic Framework 2020-2030. Initiatives like this one in the Phytosanitary Awareness Week or the current webinar series in the context of the IYPH, I think are essential to raise awareness of the importance of plant health and its contribution to food security, biodiversity conservation, and trade. In fact, we are very pleased that this initiative takes place during the extended IYPH, whose objectives are very relevant and include basically raising public awareness of the importance of plant health to achieve the SDGs and for daily life. Second, increase resources dedicated to plant health. And finally, promote good practices, knowledge, research, and partnerships. I think that continuing this open dialogue among regulators, research, and the private sector beyond the International Year of Plant Health is an essential legacy to ensure continuity of our efforts and to continue building global momentum for plant health despite the current pandemic. Thank you.
Excellent. Thanks a lot, Mirko, for this very nice remarks, and also emphasizing the IPPC efforts, especially through ISPMs, and providing global standards for movement of plant material around the world, and the relevance of ISPM 36 and 38 for moving planting material for planting that includes even seed. I think the last point on continuing the dialogue is very, very critical, because that dialogue is the one which is going to be highly relevant in the changing scenarios of policies and the pest out dynamics. So I will get back to you again later in this session. From here on, I will move on to invite Dr. Charlotte Lusty, who is the head of programs and CGIAR Gene Bank Platform Coordinator. She's based at Crop Trust, Bonn, Germany. She has a dual hat as a Crop Trust member as well as member of CGIAR. Crop Trust is an organization which provides policy frameworks and financial support to global gene banks. And I would ask Charlotte to provide her opening remarks.
Thank you very much, Lava, and greetings from Bonn in Germany to everybody. It's very nice to be part of this event. Thank you very much for inviting me and to the organizers. My particular focus then in representing both the Crop Trust and the CGIAR Gene Bank platform is to highlight the vital importance of germplasm health units and processes, not just in the movement of materials across borders, but in the actual methods of conserving germplasm, especially in the long term. So there are obvious points at which germplasm health units have a key role in the process of bringing materials into conservation. And that, I think, is fairly obvious in the collecting missions, in the acquiring of materials, and in the distribution of materials. There are obvious points where gene banks will interact with germplasm health units and with MPPOs. But perhaps what is overlooked and often is underestimated is the need also for interventions in the monitoring of accessions in long-term storage. At any point, if the viability of materials goes below thresholds, this is a moment when materials have to be planted out in the field, and in the case of tissue culture, rejuvenated also in the field. And that leads to effectively starting the whole gene bank process again from scratch, as if you're acquiring new material. So the intervention of efficient, speedy, and well-managed processes to ensure that gene banks can make sure that they're keeping their collections healthy is really very key. And this is becoming even more important if we start to envisage how the gene bank and the global system of gene banks is evolving. New technologies are coming up. We've had 30 years of research into cryopreservation, and now we're getting to a point where large-scale cryopreservation might be possible. And that's a really important point for the clonal propagated, the clonal crops, the vegetative propagated crops. This is a point where hubs can be set up to cryopreserve large amounts of very vulnerable accessions that are up until now having to be kept in the field or in tissue culture. And so we can see a much more sophisticated global system is starting to appear and surely will be developing over the coming years. And that more globally interactive system will demand yet more efficient GHU processes. And I think we should be looking at what will eventually become a very multi-layered system of germplasm health testing at all of these different levels that you can see in the slide, with real attention being given to the risks at each level. So what you might do in moving stuff intercontinentally is quite different from what you might need at a local level. And really, GHUs and all of the processes that are out there need to start organizing themselves in these kinds of layers. Capacity building is something that is really vitally important, and this kind of investment in our GHUs is critical in the coming years. Lava, I will leave it there for now. I know you're coming back to me, but if I've missed anything, please do prompt me.
Excellent. Thanks a lot. I mean, I think you said the right things in the very short 3 minutes. And actually, you said quite a lot in terms of the requirements for the global movement of the valuable germplasm and the emphasis on the capacity building. The aspect of capacity building cannot be underestimated, both in gene banks to protect the health of the germplasm, as well as the phytosanitary systems as such, especially in the developing countries, which is quite often at the receiving end of the pest outbreaks because of their inability to prevent or to mount sufficient response in right time, so that the outbreaks can be contained at the nip of at the beginning itself. So I'll get back to you again with questions coming both from the floor as well as the panel from ourselves. I would now call Tunde to post the poll. We have a question for the participants. So all you have to do is you will see a question on the screen, and then there are options. You just choose what do you think is the right option. Tunde, post the poll, please.
Yeah, thanks.
Yeah, so please, you have 45 seconds. Participants, you can choose any answer, any options that are present on the screen. And the results will be displayed soon after. So it's capacity building, more diagnostics, coordination.
Okay, so
these are all going together.
It's 45 seconds already, and we have 50% people who have voted.
Excellent. So I think we had 50%. Can they continue to vote afterwards also?
No, I can just stop it now.
Yeah, so I think we have a kind of a two close winners here based on the opinion from the participants. 50% of the participants develop better and cheaper diagnostic methods and new diagnostics for emerging pests and diseases. And the lead, the top 52% of the vote is for the coordinate monitoring and surveillance of pest and disease at a regional scale to identify emerging threats. And then the third priorities like build capacity in plant health inspection and diagnostics. So I think the trend that we see on the screen kind of reflects what the previous two speakers emphasized. So this actually forms a kind of a good start to invite another experienced panelist, Dr. Ravi Ketarpal, who is an executive secretary at Asia Pacific Association of Agriculture Research Institutions based in Bangkok, Thailand. And formerly he was a director of CABI South Asia. And before that, he was head of plant quarantine at NBPGR, which is the nodal agency for monitoring phytosanitary issues in the germplasm, going out and coming into country. And he's also one of the key members of the IPPC phytosanitary capacity evaluation tool that was developed a few years ago by FAO. So Dr. Ravi, you have 3 minutes to provide your opening remarks. We can't hear you, Dr. Ravi.
Thank you, Lava. At the outset, let me thank CG for really inviting me, and special thanks to John Cruz and Lava Kumar for that. And also, I would like to congratulate you for highlighting this issue in the International Year of Plant Health, as Marco also mentioned, which has been a sort of a neglected issue at the policy level, which is most crucial right now. Okay, in my brief remarks, what I would like to say is, to begin with, we need to be very clear of the facts. And what I mentioned here, plant health vis-a-vis germplasm health vis-a-vis seed health. There's a lot of generalities which are used, lot of confusion which goes on everywhere. We talk of phytosanitation. When we talk of phytosanitation, only plant health is included by IPPC. If they don't cover germplasm as a plant from the angle in IPPC. So phytosanitation relates to plant health. Seed is a part of plant and hence of phytosanitation that we all know. All plant health issues are not found in the seed. This we all should be on the same page before we move ahead. And secondly, plant health is important for quality seed production when it comes to seed. Seed health is important for trade and seed production, of course, also. Whereas germplasm health is as important as plant health, but in a different perspective. Germplasm seed health is as important as general seed health, but again, in an entirely different perspective, which we'll see in the panel discussion. And the risk mitigation approaches for plant health and seed health are not necessarily same as germplasm health and germplasm seed health, which will be very visible at the end of this panel discussion. Now, globally, what I see, there is an absolute neglect of germplasm health by those who make policies and gene bank standards, which Marco briefly mentioned in a very polite way, that it talks of management of gene bank and so on and all. Unfortunately, the people who authored the gene bank standard did not even think of germplasm health in the germplasm exchange or movement, which is so crucial. And we know the importance when we know how the COVID, in case of human health, is causing the issues globally. Germplasm can be an atom bomb if virulent pathogens or pests spread. And germplasm are known to carry most of the unknown biotypes, strains, and isolate, which if you compare even in publications, you don't find in normal seed health.
Yeah, that is
the operational aspects of germplasm health methodology and all are very well known, but still there is a delay globally in the germplasm transfer, which is maybe because of access and manufacturing issues or the other Nagoya protocols and all, but there are needs to expedite. And what we need right now, what we need is best practices in germplasm movement, where CGIAR has to work closely with IPPC, FAO, and National Agriculture Research Center, not to speak of NPPOs alone. So the CGIAR has come with a green path solution, which I may talk later on, which seems to be one of the most viable options. And with this, I would stop here, Lava.
Thank you very much, Dr. Ravi. I think you hit very, very important points. I mean, these are key points which are kind of, we can consider as a gaps to address the broader issues of the germplasm health policy and regulations around the world, especially when it comes to the international movement of plant health. Earlier in the introductory remarks, Jan mentioned the lack of standards for the wild species and underutilized crops. That is partly linked with insufficient knowledge about the pests and diseases. And so there are a lot of questions, and I think with the emerging demand for increasing movement of germplasm, it is the time appropriate authorities take put due attention to this matter and formulate appropriate protocols. And an attempt that CG has made is what we call as Green Pass, which we'll discuss a bit later. I would now ask Tunde to display the second poll. Okay, now participants, you have 45 seconds to choose among these options. Okay, so variable standards is a challenge. Different phytosanitary demands, different importing countries. That's number one, which is also emphasized by Dr. Ravi. Variable standards and different phytosanitary demands from different importing countries is the top constraint, 44%, followed by insufficient phytosanitary standards and insufficient diagnostic methods and insufficient capacity, complicated or inefficient procedures for phytosanitary permits or certificates. So all these are very important issues, and which were also highlighted by Dr. Ravi and also felt by the participants. So I hope we'll have some time to discuss these issues in due course. I would now invite Dr. Peter from European and Mediterranean Plant Production Organization. Dr. Peter works at EPO, and she's the lead for the diagnostics and risk analysis program. And she's also the head of XF Actors and Valley Test projects that are ongoing. Dr. Peter, you have 3 minutes.
Unmute yourself is the most famous sentence of the time. So,
We can't hear you well. Can you adjust your microphone, please?
Can you hear me now?
Somewhat better. Yes,
Yeah.
please go ahead.
Yeah. So thank you very much. I want to thank the organizers for inviting us to present what we think are the challenges with germplasm health and movement. So I will start with what is, from our perspective in the European and Mediterranean Plant Protection Organization, what are the big challenges? You have to know that in EPO, we do pest risk analysis to identify potential threats for our region, and that includes the movement from plants for planting. But our risk analyses are mainly done in the framework of movement of commercial consignments, and so big lots, etc. And clearly, when we come to germplasm exchanges, it's a different story. So the challenges that are posed by this germplasm transboundary movements are the small size. So you deal with small size usually, a huge diversity of species, and a huge diversity of potential plant pests that are associated. And on the top of it, when you send your germplasm abroad, this is really, you have germplasm coming from different origins, and then you have to distribute them to different destinations. And I can clearly see in the poll that has been done just before that this has been identified as one of the key problems. So, because indeed, when you send material to an importing country, the different importing countries have different phytosanitary requirements, which is justified because what is a quarantine pest for Europe is certainly a common pest for another part of the world. So not all the quarantine pests are quarantine pests for everybody. So how can we try to approach it? Solve is probably a little bit too strong, but if we think together, I think nothing is impossible. So as Mirko was highlighting, there are already some provision, for example, for seeds in ISPM 38, which is a standard dealing with movement of seeds. There are also other standards that are relevant, which is that instead of a lot per lot reasoning, a place of production reasoning may be more efficient. And this is where discussion with the National Plant Protection Organization may take place to have some sort of equivalence and discussion that where I produce my germplasm, I have this and this quality system in place. I'm having a regular monitoring. So I think I can give you assurance regarding the pests. So this type of reasoning are really important. So search recognition of equivalence. Regarding testing, you could, there is a difference between the target testing and the non-target testing. And we are now having new techniques that are coming
20 seconds.
So, and collaboration and international standard, but that has already been said by Mirko. So where could research help? So we have high throughput sequencing, that is an option. The generic tests that are possible for group of pests, sanitation methods, can research help there, characterization for resistance, and as Charlotte mentioned, conservation methods. And maybe more, so maybe you have other bright ideas. So these are some ideas put on the table here.
Thank you very much. Thanks a lot. We will now quickly move on to the last poll today. is the question, which is the main challenge opportunity with non-target testing? For example, high throughput sequencing. So you have 45 seconds to choose. We have about 40% voted now. Last 10 seconds, 5 seconds. Great. So we have the top choice. It's a great opportunity to discover new potential threats and prevent them before they occur. And at the same time, there is also concerns expressed. It's actually a tie. So it's a useful tool to discover unknown pathogens well ahead. And likewise, it is technically challenging for most countries to implement. So I think this is a kind of, for most modern technologies, it takes a while before it penetrates and be used by all stakeholders. But that is where the issue of capacity development will come in. But yeah, I think this is very good feedback. I will now invite our last panelist, Dr. Vivian Polar from CGIAR RTB program. She's a senior gender and innovation specialist at CGIAR research program on roots, tubers, and bananas. She's based at Lima, Peru. Dr. Polar, it's your time for opening remarks.
Thank you very much, Lava, and thank you to all the organizers for inviting me and in opening up this space for a discussion on how we can also incorporate gender in these processes. So gender and social inclusion issues are present in every area of work and research. And in the case of germplasm health units, we see that its action areas can have strong influence and also be influenced by gender and social inclusion issues. We know that germplasm moves through people and not by itself. So we need to understand the people dimension of germplasm health and pest movement. So in research and operations, some potential areas of work for the future could include collecting social and gender relevant data to inform surveillance, pest risk assessment, as I've said. Germplasm and pests move also through the actions of people that move material, and also understanding the gender roles and norms, the culture behind the processes that are linked to germplasm management and flow in every specific context. We know that women, men have different mechanisms of sharing, exchanging material, moving it from one area to another. For example, we have women who have less access to the market. They don't necessarily buy material, but they exchange it, they share it. Also, there are cultural practices associated with seed. Different indigenous people have different cultural practices related to seed management and exchange and sharing. And that has to be understood in depth in order to generate alternatives that bridge this gender and social inclusion gap, but also to inform policies, as Ravi nicely mentioned. He said policies are this area that are less explored at the moment. And we need to inform policies and regulations to address gender-related barriers and cultural-related issues that can be more manageable at the local level. This way, we can also strengthen individual and collective agency of women and indigenous people that are the ones that manage and use the seed locally and in different contexts are strongly affected also by pest pathogen spread. In the area of, in addition to work and operations, we also have the workplace. Well, Safa very nicely mentioned that there's an important gender balance, a positive gender balance in the gender or in the GHUs. And that is important to be a role model and to create awareness and foster an enabling environment. So the international centers are here also to create and build the society we want to see. And we need to act as role models to build multidisciplinary capacities of male and female scientists, and creating awareness of the importance of this cultural and gender issues to strengthen women's voice that will eventually contribute to more inclusive actions in the GHUs. Thank you, Lava. I think you're muted, Lava.
Sorry. Thanks a lot. Thanks for the very nice message. And I think you highlighted the very important aspect of informal germplasm flows that happens within and between communities, even between the countries, especially in developing world. So this is another area that needs focus, because germplasm transfers do happen outside the formal areas, and any neglect in informal sector could also be a pathway for pest spread. We definitely have to focus on these areas to strengthen the communities, to improve the seed health aspects. Okay, so now we have time for reflecting on some of these key points through some questions. I would start with the first question to Dr.
1 second, we have a last chat and then we go into your session.
Oh, sorry, yes, there is one more.
Yeah, very quickly.
Yes. I'm sorry, Peter.
Okay. No
Yes,
problem.
we have a questions to stimulate discussion on the points that was highlighted by Dr. Polar. You can read the question: which actions is required to improve gender and inclusion in access to healthy plant germplasm? So you can reflect your views in the chat box. This is not a poll. And you can continue to do.
So please, please post the questions. It should be a wave like last time. Yeah, maybe you need to post as well. But let's take 1 minute, half a minute for this, and then you have 15 minutes for your follow-up conversations.
Yes.
So I'm hoping we have 250 people here. We go. Training, very good. Capacity building. We should be getting 200 of these. Yeah, we should be. See how it goes. We give it another 20 seconds or so, Lava, then I hand back to you.
Yep.
These are actions, please, actions. Vivian, what are you seeing in the list?
Which action is required?
Yeah, what are we seeing here? A lot of capacity building going on.
Yes, I see a lot of capacity building, awareness development, and I fully agree, because many times scientists who are working at the GHUs are not really aware of the social issues. So we need to work together here with social scientists that can help create awareness and understand what are the entry points between social sciences and how that can contribute to the work of the GHUs. Beautiful, yes, the role of men and women, and a lot of capacity building. Sorry.
Yeah, that's good. Okay, now I'm going to stop sharing my slides and I'm going to hand it back to you.
Yes. Yeah, thank you. Yeah, we encourage participants to keep sharing their views, because as mentioned by Peter earlier, we are all capturing all these points through recording, and then we will make a synthesis of it. Excellent. So now we will have some time to reflect on some of the key issues mentioned by the panelists. I would get back to the panel members once again with the questions. I would start with the first panelist, Mr. Mirko. So I have two questions for you. The first one is, we had International Year of Plant Health, which is now being extended in 2021, and also for weird coincidence, the year was taken over by a human virus, COVID-19, which are kind of mined the messaging aspect, but nevertheless, the world has learned the importance of containing the spread of diseases. So in this context, I would like to ask two questions. Number one, what is being done by FAO and IPPC with regard to the delivery of international plant health as the global nodal organization? And then the second aspect is, what is one important lesson we could learn from COVID-19 pandemic for adoption to improve international agriculture?
Thanks very much, Lava. And these are interesting questions. I'll try to be concise in my response. So first of all, FAO and the IPPC Secretariat and community are working hard to ensure that plant health is given global attention in the international arena. With the International Year of Plant Health 2020, that has been extended until the 1st of July 2021, with this closing ceremony taking place precisely on that date. We are organizing a series of initiatives, and we are coordinating various efforts at the global, regional, and national level. So one of the things that we are doing is precisely ensure that people and policymakers do have the capacity and understanding to deal with plant health related issues. And this wide understanding of the importance of plant health for daily lives, but also for policy making, is certainly at the core of this whole initiative. Various different types of things were organized throughout the year. Let me just mention here that the IYPH website is one central repository of those initiatives. We counted to date 630 or plus events being organized in the various regions of the world. We also have various awareness raising initiatives targeting different stakeholders, ranging from the private sector to youth and regulators, etc. So the IYPH website is a good repository of that information, and you're welcome to consult it for taking part in this global initiative. In terms of efforts by the community, to start with, in a few weeks from now, we will have the 15th session of the Commission on Phytosanitary Measures, CPM 15, that was postponed due to the COVID-19 pandemic. And in that occasion, national plant protection organizations, together with partner organizations, will discuss the state of plant protection around the world, adopt new international standards for phytosanitary measures, and maybe even more importantly this year, they will adopt the new IPPC Strategic Framework 2020-2030. Now, this new framework is a sort of guideline that the international phytosanitary community gave itself for priority areas in the next decade, while being focused on the three let's say thematic areas that are typical of our work that relate to food security, biodiversity and environment conservation, and safe trade. The new framework also puts forward a series of new development agendas which relate to work that needs to be done by the phytosanitary community at all levels in order to continue being relevant, right? And to continue taking appropriate action to secure that plants are protected at all levels. So among these is research coordination or research networks, and the importance also of connecting laboratory networks for testing and diagnostics and so on. We also have new development agendas for the international phytosanitary certification done electronically, in other words, ePhyto. We have commodity and pathway standards that will become increasingly relevant for the future work of the CPM. So again, lots of different initiatives that can be put together and worked on collaboratively, not only by MPPOs and the IPPC world, but also with relevant research and academia organizations. You asked me about COVID-19 and its impacts on climate change, on plant health, and that is certainly something that one thing that we see is certainly that food chains could have suffered from the outbreak of such a pandemic. But we were glad that this wasn't the case so far, so much in many countries. Some others actually are at risk. Some countries and areas in particular are at risk of food chain supply disruptions. And this is the utmost concern of FAO and the international community, to avoid that we waste all the efforts that have been made in the past decades to achieve zero hunger. If there is one lesson that we can draw from this COVID-19 situation, I think I would definitely go with the one that Dr. Kumari highlighted in her intervention, which is prevention is better than cure. So we need to ensure that we put in place all the mechanisms that can sustain plant health and ensure that the introduction and spread of harmful pests can be avoided in the first place before dealing with their management. Thank you, and over for now.
You're on mute, Lava.
Thanks. Yeah, thank you. These are very important issues, and we wish we had more time to discuss these topics. And I'm very glad to hear that the new strategic framework is going to be released in the 15th CPM, and it is going to have a provision for research networks. I strongly believe CG centers have a big role to play in that, because of our strategic positioning and the kind of work that we are doing, especially with regard to the phytosanitary testing of the germplasm. In this context, I would like to ask a question to Dr. Ravi. You highlighted a number of issues with regard to the gaps in the phytosanitary policy and regulations when it comes to germplasm health, and lack of distinction between the seed and the germplasm. What do you think should be prioritized in a kind of a immediate action to make a change?
Thank you for the question, Lava. If you look at the germplasm as such, it's a baby of agrobiodiversity or conventional biological diversity. And if you look at CBD is completely silent about phytosanitation aspect. We only talk of cryopreservation and ex situ conservation, access and benefit sharing, and so on. And the world finishes there for CBD. If you look at the plant and commercial consignment, there's WTO, which has a standard setting bodies like IPPC, OIE, Codex, which looks at the big consignments. The germplasm is, of course, a research consignment, often having a small sample size. So there is a big problem at the top level. We really need to have a top-down approach here. There are no guidelines for safe movement of germplasm. International Plant Genetic Resource Institute, re-christened as a Biodiversity International, they developed safe movement of germplasm guidelines for about 17 or 18 crop species. Excellent piece of work. About 14 years back, they stopped doing it. I asked the then Director General, why is it so? He said, we have no funding. It means funding is actually showing the importance of the subject. What I want to say, when biodiversity failed to highlight the importance of biodiversity, because by Biodiversity International, then what do you expect more? So there's a complete neglect. Now, in the light of this, the whole germplasm quarantine in the movement, transboundary movement, becomes NPPO specific. Neither you can have SPS standards, which are large consignment, there is sampling, there are so many things. And NPPOs are not bound by ISPNs, which are mandatory. The application of ISPNs in terms of risk analysis and all, which is a big gap. There are some disconnected, half-hearted efforts made by NPPOs, but some countries have done quite well on the ground level, like India, I should say, where I happen to work, where they have designated a research organization, germplasm being a research material, in the legislation to look at the germplasm quarantine. So there is a comprehension, understanding, appreciation of germplasm movement. For the NPPOs as such, who deal with millions of metric ton, if you give them 50 grams of germplasm, they say, hey, what's this? Okay, let it go. You know, like those kind of approach. So globally, we are in a quite disconnected position. And my point here is, if we want to hit a bullseye, we have been talking about germplasm health in transboundary movement with all evidences, examples, and all. It is a time we need to come to an action plan. Thanks to the first action plan I could see was when I found the GHUs of CGIAR aligning. The alignment of the GHU of CG system was the number one step. Otherwise, CG was working in isolation. We had CIMIC quarantine, we had ICARDA, we had ICRISAC, and so on. Now you are aligned, and your publication, which I saw recently, I think is a masterpiece, which we all should factor and see like what are the phytosanitary implications on safe global movement of germplasm. The message there, which we get clearly, it's like it will be like a Bible for years to come. That given publication, which was shown in the chat, and I recommend everyone to go through it critically, because now the system we have been talking about of a green pass, based on the checks in phytosanitation of the germplasm, in the flow of germplasm, right from exploration, collection, conservation, characterization, evaluation. There have been checks at various levels. All that pulled together, well done, well data made, really is a good subject of qualifying it to get a green pass, which is your terminology, Lava, and we really appreciate that. And the green pass thing has, when we talked to FAO a couple of years back, IPPC, they said there can be no standards. ISPM 38 does not relate to germplasm at all. Only 10% is applicable. Where do we come now? We have to go for a best practices. IPPC is doing a lot of best practices. So we need to go for best practices for germplasm exchange is number one in the action plan. Secondly, to do that, IPPC will not come and do it for you. IPPC is a group of a handful of people with too much of work, but they have norms. You need to approach them through NPPOs. And this year being International Year of Plant Health, you need to mobilize the NPPOs, make an agenda for the IPPC, and also you need to, being a 1CG now, you have a heavier voice, a much better voice. I think it is from that management role of CG, it should not go to IPPC directly. CGIAR should approach FAO, because IPPC is WTO standard making organization. That is for commercial consignment. FAO should take up this agenda of IPPC, because IPPC secretariat is in FAO building. So you need to be very smart in your approach. Through FAO, CG, attack IPPC, through NPPOs, attack IPPC, and make sure that this is accepted as a model case for best practices for CG germplasm. And finally, your challenge will remain how to bring in the National Agriculture Research System and all other actors in the whole show. So I think Green Pass is a very good, viable solution. We have had a lot of academic discussion. It is a time for an action plan, which should come as a recommendation of this webinar. Otherwise, we again have another academic webinar, and that is it. We have been having it, even on germplasm health. So I think we should mean our business. I'm sorry to be a bit blunt, but I really want this subject to be pushed forward and through the CGIAR group people, and it merits to be at the global front. Thank you.
Thank you. Thanks a lot, Dr. Ravi. I mean, I could not. I mean, these you made excellent points and excellent suggestions with regard to how to move forward with regard to the addressing key gaps with regard to the international movement of germplasm and the phytosanitary health issues, and some of the solutions that you have provided, how to approach FAO through the various stakeholders, especially NPPOs, are very critical. And I would definitely take up. Mirko is here as representing both IPPC and FAO, and he's keenly listening what we are talking, and I'm sure he would be considering some of these elements as part of the AOB of CPM 15, which is due at the end of March, at the beginning of April. So we don't have much time. I will have to quickly call on with one more question, very important question, to Dr. Waiter from EPO. You mentioned the importance of technology and diagnostics in addressing the challenges. And in the poll, for the question that we asked on the use of generic diagnostics such as NGS, people do feel the relevance of the tool, but at the same time, the capacity is an issue and interpreting the results is an issue. What is your experience in EPO with regard to the use of NGS-based diagnostics? And how do you see it as its global role in a 60-second response, Dr. Peter?
I'm going to try to be short on that. So HTS, we are trying to develop guidelines because there is all the question about validation, interpretation, which indeed is tricky. So we are at the moment trying to develop some guidance about that. So how you could validate and the HTS technology. This is a very evolving tool at the moment, and the cost at the moment, the cost is going down and down and down. And I think Jan is better expert than myself to comment on that issue. So I think it's going to be a tool that's going to be available in the future to more people with the smaller technologies. I think yes, I think there are still issues and we need to harmonize that. And I would like to draw your attention to an FAO and IPPC document on the use of HTS technology, which Mirko can probably give the link to in the chat. So there has been some discussion on the issue of use of HTS, because one of the issues is what is the relevance of what you find with HTS. And that's also a critical point. And again, another issue, I don't want to open a Pandora box, but when you detect DNA, that's not a proof of the living organism. And that's another issue that research will have to look at collectively. How can we use indirect tests in the future? So, and I can only support what Javi has been saying. Discussion and interaction between the different bodies that are dealing with safe movement of germplasm is essential to come up with a global solution.
Thank you. I think there are 2 points. It's very important to not only just results, but to look at what the results are saying with regard to the knowledge on the pathogens that are available. And thanks for also alluding to this IPPC document on interpreting HTS results. I think that's an ongoing research, but sure, that is a tool for the future, and we probably have to embrace and adopt as part of the routine phytosanitary diagnostics. The last 5 minutes left.
For nidification, for propagation material, that's really becoming more and more commonly used.
Yeah. Okay. So I have one question to Dr. Lusty, Charlotte Lusty. What are the most important actions to ensure we deliver benefits of healthier plant germplasm to farmers? So if you have 60 seconds response, please.
Oh my goodness. Well, I think again, I return to the point that you have to look at the risks and align your practices with the risks. That's something, Lava, that you've taught me very well. So I think building a system that suits the risk, that suits the crop, that suits the objectives is important. And I think we've been working far too long relying on MPPOs to do everything. And I think now we need to look, as Dr. Ravi's been saying, about a much more built-up system with support coming from higher up for activities that go beyond just movement across borders, but really touch upon the conservation of these and that the long-term collaborations that we want to have around these resources into the future. So I feel very attuned with what's been expressed already, and grateful to hear it.
Thanks, Sue. Thank you. I think that's a very, very good point. It's time for action, and understand the risks are around there. We need to put what we are saying into practice. So I think that's where people like you have a big role to play with your international connections. The last question to our Dr. Polar. Which actions will help us overcome gender and inclusion challenges in improving access to healthy plant germplasm? Would you please respond? I know there is a lot of chat in the chat. There was a lot of commentary, but I hope you had time to quickly check and synthesize it in 60 seconds. Dr. Vivian.
Thank you, Lava. Yes, there was a lot in the chat, and actually, I think which actions would help us has to do a lot with capacity development and awareness creation. We see that many times it is difficult for the specific technical area to understand how to address issues of gender and inclusion. And it requires a thorough dialogue with social scientists to understand how people's cultural background, practices, norms, roles of men and women and indigenous populations are connected, influence our work, and are in return influenced by our work. So the area of surveillance and pest risk assessment would benefit very much from this internal dialogue between social scientists and biophysical. Thank you, Lava.
Thank you. Thanks a lot. With this, we will conclude the panel session. I thank you all of you for excellent insights. I think you have given a lot of ideas and a lot of food for thoughts. So I think we have a lot to reflect following this the webinar. There are a lot of key takeaways. I mean, we can actually synthesize. There are at least 10 points, but I will just make into kind of a 5. The key areas that we need to focus. Number 1 is to maintain the dialogue among the stakeholders. International plant health is a multi-stakeholder issue, and we need to, and it operates at different levels, and we need to continue dialogue at all angles. The capacity development has come up in a number of areas. I think we can't underestimate the value of capacity development. And the third aspect is there are a number of gaps have been identified. That is a good thing. We know what needs to be done. And we all probably also know what is the best way forward. And what is missing is action. And that is something that we need to do. Action to address the key gaps. The last aspect is the technology. We can't underestimate the value of technology to address the phytosanitary challenges. We can have all the solutions, but if we don't know how to tackle them, then it is also a problem. So with all the new technologies that are coming up, we need to mold into to fit for purpose for the use in the phytosanitary context. And the last aspect is not to neglect the inclusion aspect. Germplasm flows also happens at the community level, and it is very, very important we pay attention to that sector as well. So with that, I will hand over back to Peter.
Thank you very much, Lava. A really good session. We got a really rich set of ideas and feedback and input and lots of food for thought. Right, we're going to do a quick closing before you rush off. We're going to take 3 more minutes of your time, and we're going to do one last chat for you. We'll be talking about solutions and actions. So what is the most promising solution or action we need to take to achieve plant germplasm health? Speakers, please also put your comments in here. Yeah, so let's have the same. Before you say thank you very much and goodbye, give us your final parting words. Tell us, what's the most promising solution or action we need to take? Is it influencing the IPPC? Is it going to the CBD? Is it reaching farmers? What is the most action? Is it empowering women? What is the most promising solution or action we need to take? Is it better diagnostic tools? Please post something in the chat, please. Someone's asking me about the seminar presentations. They will be shared. There will be a video presentation, I think, shared of all of this. Thank you very much. But please put the text in there. Green paths for seeing, collaboration, linkages, working together, cooperation. What are we seeing? What's the most important, the most promising action? the most promising action plans. Thank you, Ravi. That looks like a different Ravi. Okay, global collaboration, special regulations. Yes, okay. So please keep on posting very quickly your messages, and then we're going to wrap this up. So while you post the messages, tell us what the most promising actions are. Let me just advertise the next 2 webinars. We have 2 more coming up. One is on the 10th of March about integrated pests and disease management. And and then the final one is the 31st of March, and it will be looking at One Health. So those 2 are still available. They're being planned at this moment. They're looking very exciting, and they will continue the banner from today. Right, I would like to hand over to Jan. I think, 1 second, while Jan is turning on his video and his audio, just advertising this publication that came off the press, I think only a few days ago. It covers exactly the topic of today. So I'm assuming every question, Lava, that we were asking is answered in this paper, right? I'm not hearing you.
More details can be found in that one.
Yeah, so this is the paper if you want to have all the answers. Jan, are you there?
I'm here. Do you hear me?
Super, I'm hearing you perfectly. So your job is the last slides.
Yes, indeed.
And any final thoughts you have?
Yes, indeed. Well, I would like to close this session with giving a word of appreciation to you, the audience. I think we've had a lot of interaction. There's been a lot of feedback in the chat, and it has been really enriching, I think. It will take us some time to analyze this and see what we have learned from this. Also, special thanks to our speakers for sharing their valuable time, experience, and knowledge with us. The discussion group has been really great. I would like to thank our moderators, Lava and Peter, for managing and stimulating discussion and keeping us more or less on time. Also to our organizers, Lava, Peter, James Stapleton, who was behind the scenes, Nina Ibabao, myself. And also our communications and Zoom team, Peter, James, Nina, Viviana, Katos, Catherine Lopez, and Tunde, who was managing the Zoom session. And of course, next slide, please, to all our partners and sponsors of this event. And we really look forward to seeing you in the next seminars. I think, like Ravi and many other people said here, we need to convert what we have discussed in this seminar into some action, into an action plan, how we can actually move the things forward and get some traction on how to deal with the germplasm health, and not just talk. We need to walk the talk as well. So with that, I think I will declare this session closed and thank everyone again once more for their participation, and hope to see you next time. Bye, everyone.
Bye.
Great. Thank you, Jan.
Thank
Bye,
you.
everybody. That was excellent.
Do you have a photo session, Lava?
Yes. Oh, I think there have been photos being taken throughout, so we will share.
Thank you. No issues.
Yeah. I think we should have done it before, but I'm sure our Zoom person must have been taking pictures. Thanks a lot for all your excellent advices. We have a lot to think and digest.
Oh yes, this was great.
Thank you.
Yeah.
Thank you for the opportunity to participate. Thank you all the organizers.
Yeah, and the people like you with all your connections, and especially with the membership network that you have among the national programs. I think you probably have to do a lot with regard to mobilizing the stakeholders so that we can pitch in this to IPPC.
Sure.
The only thing I wanted to, I didn't want to say anything, but I think there are some timelines. Oh, Mikko is there. I couldn't see you anymore, Mikko. But topic suggestion, for example, is still open.
It comes to